Jonathan Plett. I am a researcher here at Hawkesbury Institute for the Environment. So my area of expertise and interest is looking at how microbes, so bacteria and fungi, communicate with plants. That might sound a bit interesting from the aspect of pathogen detection, but it's one of the things that we are realizing is that for pathogens to overcome a plant and cause disease, they actually release a lot of proteins that we would call communication tools, in which they actually overcome the plant and overcome its immunity. So that area is the specialty that I'm bringing into this project to understand not only how we can detect pathogens, but how we can also treat them and get rid of them, hopefully from a grower's area. So there's a number of areas that I've been very lucky to work on over my career. A lot of them, though, I think the area that kind of glues them together is what I'm really interested in, is the ability to take different plants and microbes that a lot of people have not been able to study, what we would say at the genomic or genetic level, and provide the tools that now the whole area of science can begin looking at that. So I've been looking at different beneficial fungi and looking at genetics in them and how they colonize plants. In this project, it's been really cool with Aastha to be able to develop the very first genome of one of the isolates of this pathogen within Australia. And that is giving us really great insight into how it's overcoming the host and how we might also, again, leverage that to help the growers.
I'm Aastha Ranjan and I am a PhD student in the Western Sydney Uni, working under the supervision of Dr. Jonathan Plett in the HIE, Hawkesbury Institute for the Environment. My PhD project is about controlling the gummy stem blight. It's a fungal disease in the glasshouse cucurbits, and my crop of interest is cucumber, which is specifically baby cucumber, sold under the name of Cukes by Perfection Fresh in the market, and I think everybody loves to have them as a snack. But what people have found out that when the growers harvest those cucumber, then there is no disease or symptoms of the gummy stem blight. But when it goes into the hands of the consumers, they found out that it is diseased or the cucumber has been spoiled. So it's basically a post-harvest loss problem for the growers. So here we are trying to develop early detection tools which can be used by the growers to detect the pathogen even before the symptoms get developed in the cucumber. We are also trying to differentiate the gummy stem blight and other pathogen so that they can use the fungicide specific to the gummy stem blight instead of the fungicide which they use for other pathogen, because sometimes which work for one pathogen does not work for gummy stem blight. So we are trying to develop molecular tools through which we can differentiate between the fungicides, which is helpful for the gummy stem blight.
This project is supported by Future Food Systems. So we were approached by Perfection Fresh to talk about if we could use some of our specialized knowledge in how microbes interact with plants to be able to help them with early detection. So one of the main areas that they have a problem with is that they really want to be able to reduce their environmental footprint. They want to be able to treat with less fungicides, they want to be able to treat earlier, and they want to be able to produce the best quality products that they can for their consumers. So one of the challenges we've found in interacting with any of our industry partners through Future Food Systems has been the fact that they want to be able to do better at surveying for disease. There are companies that can do that for them, but we are finding that that cost is something that is very hard for industry to overcome. And so they are not able to survey as much as they would like. So the challenge we're trying to overcome here is trying to develop very cost-effective means of being able to detect pathogens. And that's not only going to help industry here, but our longer-term outlook for that is really trying to develop something that is so low cost that this is something that could even be taken up in other countries, low-income countries, where growers don't have access like we do here in Australia. And so using that technology and using the innovations that we are producing, can we then extend that further beyond Australia and help other countries?
I will be developing advanced molecular technology which will help us make the plant more resistant towards the disease and the fungal pathogen more susceptible towards the protective measures. And also there will be early detection tools which can be used by the growers on field for the detection of the disease. With the development of these advanced molecular techniques and the early detection methods, it will help the growers to do the on-field detection without the requirement of any specialized machinery or a specialized knowledge or training. Because usually while working in the lab, it requires a specific instrument or a specific equipment to work with that. But we are developing tools which would help the grower to just do a direct colorimetric test or a fluorescence test, which will help them to recognize the disease without the symptoms get developed. And also it will help them to release good cucumbers in the market, which is not only symptomless but also disease free, and it will also generate a good consumer and producer relationship.
So next steps of our research. So there's a few avenues that we are going to pursue once we have developed this to a point that is past proof of concept. This current project really is a proof of concept project. Can we use things like genomics or proteomics or other very specialized techniques like this to be able to detect these pathogens very early? We think we will, and we have some early successes that we would show that we're on the right path. To put that though into the hands of a grower, the next step is going to be then working with other companies to develop what we have into a kit that anybody can buy and anybody can use with very minimal training. Another way that we are going to amplify what we are doing is definitely we are academics. We are going to share this freely through publication. We will be going to conferences that we host or that we attend to be able to say to people, look, this is what we have produced in this Future Food Systems project in collaboration with Perfection Fresh, but this is something that we want you to try, we want you to take up. We also have collaborations with other people. We have collaborations with state facilities that their job is to do pathogen detection and pathogen protection. So we also are working with them in this project and in other projects to try and make sure that what we are producing is something that will be easily adaptable to what state facilities can also use themselves in the ongoing years.
Being in the field of science, it is all about curiosity, learning, new experiences, trying. So in this field of science, everyone's voice matters because everyone has their own ideas, has their own perspectives. If you want to do a research, you already belong here.
So one of the things that we're also doing through Future Food Systems is we're also looking into beneficial. So one of those beneficial fungi that is really critical in all ecosystems are fungi called mycorrhizal fungi. And so in July, we're actually going to be hosting in Cairns, in Queensland, the International Conference on Mycorrhiza. And this is where people from all over the world, as well as Australia, are going to be coming and they're going to be showing their cutting-edge research in mycorrhizal fungi and how we can be using that not only in ecosystems that are environmental, but also in agriculture and in forestry and in areas like truffle research, to be able to give new tools for people and new avenues of research so we can advance sustainable agricultural production.